Welcome back to The Explainer. Today, we're exploring a microscopic universe that is, quite literally, all around us. It's inside us, it's beneath our feet, it's everywhere. We are diving deep into Kingdom Monera, pulling our focus straight from the classic NCERT biology syllabus. Now, just to give you a sense of the sheer mind-boggling scale we're dealing with here, if you walk outside right now and scoop up just a handful of soil, you are holding hundreds, if not thousands of bacteria right in the palm of your hand. They are the invisible empire that actually runs our world. Okay, let's dive right into this. To really grasp Kingdom Monera, we first need to lock down the foundational concept of the entire kingdom, and that is the prokaryotic cell. A prokaryote is a single-celled organism that completely lacks a true membrane-bound nucleus and other specialized organelles. So they're simple, right? And they are incredibly ancient. But as we're going to see, that simplicity is exactly what makes them so unbelievably adaptable and successful. Here is our roadmap for today's microscopic tour. We'll kick things off with a welcome to Kingdom Monera, then look at bacterial anatomy and shapes. Next, we'll brave the extremes with Archaebacteria before meeting the true bacteria and the ultimate rule-breaker, Mycoplasma. Finally, we'll cover how they eat and reproduce, and then wrap up with their massive impact on our world. For all you focused learners following along, don't worry, we're covering all these critical exam topics super logically and clearly. Section 1. Welcome to Kingdom Monera, the sole prokaryotes. Now, what's really interesting here is seeing exactly where Monera fits into the grand scheme of life. We have to look back to R.H. Whitaker's famous 1969 five-kingdom classification system. Notice how Monera stands totally alone in that top row. Out of all five kingdoms, Protista, Fungi, Plantae, Animalia, Monera is the absolute only one with a prokaryotic cell type. They feature a non-cellulosic cell wall, and that all-important nuclear membrane is just completely absent. They're biological outliers in the best way possible.
So what really makes a Moneran a Moneran? Well, for starters, they're the oldest forms of life on Earth, and they're by far the most abundant microorganisms on the planet. They are strictly unicellular and have zero membrane-bound organelles. Their genetic material is just naked, circular DNA hanging out in a region called the nucleoid. They pack 70S ribosomes, and honestly, their rugged, stripped-down structure allows them to live in the most extreme habitats you can imagine. We're talking boiling hot springs, freezing snow, and the crushing depths of the oceans.
Moving right along to Section 2, Anatomy and Bacterial Shapes, where form truly meets function. We know they're simple, we know they're mostly invisible to the naked eye, but what does a microscopic empire actually look like? It's a great question. Because despite having such a basic internal setup, the way bacteria behave and the shapes they take on are incredibly varied. In fact, their physical shape is one of the absolute main ways scientists identify and categorize them in the lab. Let's break down the first two primary shapes. First up, we have the coccus, or plural, cocci. These are your perfectly spherical-shaped bacteria. A really common example that you've definitely heard of is Streptococcus. Yeah, the one that causes strep throat. Then we have the bacillus, or bacilli. These are rod-shaped. Picture a tiny microscopic pill or a cylinder. E. coli and Bacillus anthracis are classic textbook examples of this rod-like structure. Next, we've got the Vibrio. These are fascinating comma-shaped, kind of curved rods. You're going to find these frequently in aquatic environments, and a pretty notorious example is Vibrio cholera. And finally, we have the Spirillum, or Spirilla. Just like the name suggests, these are spiral, helical, and quite rigid in shape. They usually move around their environment using this whip-like tail called a flagellum. Remembering these four distinct shapes is absolutely crucial for categorizing any bacterial species. All right, Section 3, Archaeobacteria, the extremophiles. This is about surviving the unsurvivable. If the microscopic world had rugged sci-fi action heroes, it would absolutely be Archaebacteria. These guys have a serious knack for surviving the unsurvivable. So, the crucial point here is understanding the fundamental split between them and Eubacteria. Archaebacteria are ancient extremophiles. They've got a completely distinct cell wall structure that totally lacks peptidoglycan, and they have unique ribosomal RNA. This unique biological armor is what lets them thrive in extreme environments. On the flip side, Eubacteria are your true bacteria. They have a rigid cell wall made of peptidoglycan, and they're found pretty much everywhere else on Earth. To really give you a sense of how tough Archaebacteria are, think of them living in environments that resemble straight-up alien planets right here on Earth. Halophiles thrive in intensely salty areas like the Dead Sea. Thermoacidophiles
They don't just survive, they actively thrive in boiling, highly acidic hot springs. And methanogens, they live in strictly anaerobic, marshy areas and even in the guts of ruminant animals like cows, where they are entirely responsible for producing biogas, which is methane.
Let's move to section 4, Eubacteria and Mycoplasma, true bacteria and the ultimate rule breakers. Now, when we talk about the true bacteria that we interact with on a daily basis, we have a massive clinical distinction based on a lab test called gram staining. Gram-positive bacteria have this thick peptidoglycan layer, no outer membrane, and they stain a deep purple. Gram-negative bacteria, though, have a thin peptidoglycan layer, but they possess this sneaky outer membrane made of lipopolysaccharides. They stain pink. And here's why that matters. That outer membrane in gram-negative bacteria acts like a chemical shield, making them way tougher to treat with our traditional antibiotics. Also hanging out in this group are the Cyanobacteria, which you probably know as blue-green algae. They are incredibly important ecologically. Just like green plants, they're photosynthetic autotrophs containing chlorophyll A. You'll find them in freshwater, marine, and terrestrial environments. But what's truly amazing is that some of them, like nostoc and anabaena, have specialized cells that can literally pull nitrogen right out of the atmosphere and fix it into the soil. They are basically nature's own fertilizers. All right, now for the rule breaker, 0. That is exactly how many cell walls Mycoplasma has. 0. By completely ditching the cell wall, Mycoplasma holds the biological heavyweight title for the smallest known living cell. It's so adaptable it can survive completely without oxygen, although, fair warning, it is often quite pathogenic to plants and animals. Section 5, Nutrition and Reproduction, or as I like to call it, the art of survival. So how does this massive, invisible empire feed itself to conquer literally every corner of the globe? Well, bacteria actually show the most extensive metabolic diversity of any organism group. Some are photosynthetic autotrophs, using sunlight to make food. Shout out to Cyanobacteria. Others are chemosynthetic autotrophs. These guys play a huge role in nutrient recycling by oxidizing inorganic substances like nitrates and ammonia to create ATP energy. But keep in mind, the vast majority of bacteria are heterotrophs. They depend entirely on other organisms or dead organic matter, acting as nature's ultimate decomposers or sometimes as parasites. Let's move along and see how this builds. Once they're fed, how do they multiply so rapidly? Well, most of the time, bacteria reproduce asexually through this really rapid, elegant process called binary fission. It's just 3 steps. Step 1, the circular DNA duplicates. Step 2, the cell elongates and the duplicated DNA separates to opposite sides. Step 3, the cell literally just splits right down the middle, creating 2 identical daughter cells. Get this, under ideal conditions, this whole sequence can happen in just 20 minutes.
But of course, biology always has exceptions, right? When conditions get harsh, like really unfavorable, bacteria can form incredibly tough spores to ensure they survive until things get better. And then there's conjugation. This is a primitive form of sexual reproduction. 2 bacteria make direct cell-to-cell contact using a tiny tube called a pilus. The donor transfers a small circle of DNA, called a plasmid, over to the recipient. It's essentially a genetic superpower. It allows them to rapidly spread highly advantageous traits, like, say, antibiotic resistance, across an entire population.
Finally, Section 6, Impact on our world, the good, the bad, and the microscopic.
And this brilliantly illustrates why all this textbook biology actually matters. The real-world impacts of Monera on human life are massive. Bacteria are not just germs. They are literally the invisible pillars holding up our world. They're essential for nutrient cycling, acting as decomposers that break down dead matter so life can keep going. They drive nitrogen fixation. Bacteria like Rhizobium enrich our agricultural soils. They're masters of food production. Lactobacillus is what ferments milk into the curd and yogurt we eat every single day. And in medicine, bacteria like Streptomyces actually produce the essential antibiotics we use to fight off other bacterial infections. Of course, we absolutely have to acknowledge the dark side of Monera. Pathogenic, heterotrophic bacteria are responsible for some of the most devastating diseases in all of human history. We're talking about cholera, caused by Vibrio cholerae, typhoid from Salmonella typhi, as well as tetanus and tuberculosis. Their impact on human mortality and, frankly, on the entire development of modern medicine simply cannot be overstated. Which brings us to a final, slightly provocative thought to leave you with today. Is Kingdom Monera actually 2 distinct domains? See, in the late 1970s, a scientist named Carl Woese discovered that the genetic and biochemical differences between Archaebacteria and Eubacteria were so incredibly vast that they really shouldn't even be lumped into the same kingdom. He proposed splitting Monera into two completely separate domains, Archaea and Bacteria. It really leaves you wondering, as our microscopes get sharper and our genetic sequencing gets faster, how else will our understanding of the biological tree of life completely evolve? Thanks for joining me on this microscopic journey. Keep exploring, and I'll catch you on the next explainer.